It's my great pleasure to introduce Professor Azim Surani. Now, if ever there was a case where you would say the next speaker needs no introduction, this is probably it. But for convention, I'll go through some of the facts that I gleaned about Azim from the website. So, received his PhD from Cambridge University in 1975, joined the Babraham Institute in 1979, and discovered genomic imprinting in 1984. So that's quite a biography, isn't it? Subsequently, novel imprinted genes and their functions were studied with mechanisms through establishment and erasure of DNA methylation. He's an elective Fellow of the Royal Society, Fellow of the Academy of Medical Sciences, and was awarded a Royal Medal in 2010. So as I say, really it doesn't need any introduction. So very much looking forward to hearing him speak, and we're very grateful that he agreed to kick off the conference with this keynote address. Awesome, thank you.
Thank you very much. I really would like to thank the organizers and the rest of the organizers for inviting me to this meeting. I haven't actually been to a genomic imprinting meeting for quite some time, and it's wonderful to be here and seeing a lot of faces that I haven't seen for a while. So, what am I going to do? So, I think it's nearly 39 years since I bumped into genomic imprinting, so it's quite a long time ago. And of course, a lot has happened since then. You know, there have been lots of advances. You know, a lot of imprinted genes have been discovered. We know about the functions of some of these imprinted genes. We also know quite a bit about mechanisms of how imprints are put on and erased, and some of the consequences of imprinting or an impact on not only on development but also on disease. So there's been quite a lot of progress. So what I want to do really is to take a slightly, I think we're going to hear a lot of talks on some of these areas. So I'm going to actually take a developmental biologist's view of the field to some extent, and also to talk about the work that we've been doing on the germ cell lineage, both mouse and human. So these are some of the things that I want to sort of bring up. So one of the things that I've recently been interested in is how imprinting has had an impact on mammalian development. I think that there's a change in strategy of development, which is partly linked with imprinting. And the other thing that is interesting is that the imprinting cycle has actually got incorporated into germ cell cycle. And this is the reason why I got into the germ lineage work. So imprinting is obviously intergenerational epigenetic inheritance is essential for totipotency in mammals and development. And it's actually a really good paradigm for epigenome reprogramming in vivo. Is one of the rare cases where we can understand how the information, epigenetic information, is put on and erased. So I actually started in this quest, started with this question, actually. This was a question that I was asking. Are both parental genomes essential for development in mammals, which became kind of somewhat of an obsession? And in the end, of course, the answer is that in mammals, you do need both parental genomes. But at the time, there was a lot of debate about this, because you have some parthenogenetic vertebrates like these lizards, they can reproduce parthenogenetically. So it's not unusual that we could ask this question. And also, there was a lot of debate, and there's this one book here that was published by Jefferson Gribbin in 1984. Is sex irrelevant in the modern world? So there was quite a lot of discussion, ongoing. But a few months later, we finally published our paper to show that indeed you do need both parental genomes. And the reason is that in mammals, the parental genomes are epigenetically asymmetrical. So although they pass on equivalent genetic information, they are not epigenetically similar. So that's why you need both parental genomes. And of course, we know from the work of Bruce Katenack and Joe Peters, who's here, that there are these chromosomal regions in mouse and human that are imprinted and contain epigenetic information, which depend on the parental origin. And so we also, as I mentioned, know quite a lot of genes that are imprinted. So here are two that show expression when they come from the mother, and these are two which are expressed when they come from the father. So the information is accumulated over many years. So they show a monoallelic expression of either maternal or paternal allele. And it's a heritable change. So the mark is heritable. So it comes from the germline, it's heritable, and is transmitted through to adulthood. So what we don't know is how did the whole thing start. I think this is something which is still a bit puzzling, and I don't think we really know how imprinting actually started. I think we can look at some of the imprinted genes and maybe think about how the process could have started. PEG10 is quite an interesting gene. It's a transposon-derived neogene, and it's only present in marsupials and in eutherian mammals. It's not in other vertebrates. And it's imprinted, so the paternal allele is active. And it's critical for development of the placenta. So, and like H9-IGF2, PEG10 is amongst one of the earlier genes that's imprinted. But they seem to be important for the development of the placenta. Just to take another example, DLK-DIO imprinted domain that Anne Ferguson-Smith and some others have been working on. This is a slightly more complex domain, has several genes on the paternal genome, paternal chromosome that are expressed. And there is this imprinting region here where you get the methylation mark that's put on when this chromosome comes from the father. And it has got ZFP57 binding site, so that's the zinc finger protein that actually binds to it and propagates it through our development and later on in adulthood. And recent paper shows that if you have a mutation in this binding site, it will abrogate imprinting, and you prevent this imprinting status to transmitting to through development into adulthood. So there are mechanisms that recognize imprint and transmit them throughout different somatic tissues as the embryo and the adult develops. So the propagation of the imprint after and the impact it has on the expression of these imprinted genes is, we are familiar with, and we know that these expression of these imprinted genes is important for development, and there are lots of examples of that. And you have these two zinc finger proteins which recognize and propagate imprints to adulthood. And so there are mechanisms for the mark being put on in the germline and the propagation subsequently. So we know how the system, broadly speaking, actually works. So there have been attempts to kind of look at all the imprinted genes together. So there are this, what about the possibility that these imprinted genes could work as a kind of a network? And we were of course, when we first started doing this kind of work, we were thinking about individual imprinted genes and thinking about how imprinted gene in a particular domain has an impact on development and disease. So this is how we have actually been looking at this problem. But there may be some important aspects that we need to kind of think about, whether collectively they have some kind of a role in development, growth, physiology, behavior, metabolism. So that these are aspects that we know are affected when we mutate individual imprinted genes. And so we have, and also have some examples like that. So we have now got some idea about how the mark is acquired, transmitted, how it's recognized, and some of the genes that get imprinted and the role they may have later on. I think the toughest question has been evolution of imprinting. How did the system actually evolve? And there are a number of proposals that have been put forward. I think antagonism theory, conflict theory, I think was one of the early ones, kinship theory, co-adaptation. So there are a number of things that have been proposed. I don't think any of those individually are satisfactory, because I think we still don't really have, either we don't know how to ask the question, or we're missing something. I think what I would like to do is to kind of think about it from, as I said, developmental point of view, and because I think imprinting is linked with a fundamental change in the strategy for development amongst vertebrates. So we can think about it from that point of view and see if we can then understand why this system might have evolved. So the first thing is that we know that evolution of genomic imprinting is in mammals includes marsupials and eutherians. It's not present in monotremes, the egg-laying mammal. And so it's associated with placentation and viviparity. So this is placental viviparity is one of the areas which is connected with genomic imprinting as we understand it from a mammalian point of view. As I mentioned, it is observed in marsupials, but as far as I've been able to find out, there are currently 6 imprinted genes that have been identified in marsupials, as opposed to over 100 in eutherians, in mouse and humans. The thing that I think is also interesting from development point of view is that the marsupials have relatively short duration of development in utero. Implantation is quite late, so it's a short time during which the embryo is dependent on the placenta in marsupials. So I think one of the things I'd like to do is to sort of compare marsupial development with eutherian mammal development to see what we can find out about imprinting and how it might have evolved. So if you first look at pre-implantation development in marsupials, you can see they have this stuff called deutoplasm. It's kind of a substance which might be used for nutrition of the embryo. So unlike Eutherian eggs, they have this kind of substance here which persists during this early pre-implantation development. And you can see here, I think if you're familiar with Eutherian early development, you can see that it's not quite like what we are familiar with when you're working on mice. You have this unilaminar blastocyst. And then, as I mentioned, this late implantation, if you look at Monodelphis domestica, it's late implantation around day 10 or 12. A very short gestation period. The embryo, the birth date is around day 14. So it's very short time when it's dependent on the placenta. So I think that's kind of interesting. And I think a lot of the debate has been about how imprinting is important for placental development of the placenta and the nutrition and so on, which is obviously true. But I think the other point to make is that it probably also has quite a marked impact on the development of the early pre-implantation development. So if you, for example, look at this slide here, looking at early development in monotremes, marsupials, and eutherians, these are the monotreme embryos which are dependent on yolk for development. I also told you about marsupials. You can see the marsupial development, unilaminar blastocysts giving rise to both pluripotent cells and also trophoblast cells coming from this unilaminar blastocyst. Now, if you now look at the eutherians, here you now start to see cells that are completely enclosed during early development of the embryo. And these are the precursors of the inner cell mass. They're going to give rise to epiblast. So you now have Eutherian blastocyst, which has got distinct inner cell mass with pluripotent epiblast. And then these cells here are going to give rise to the trophoblast. So this, what this tells us is that Eutherian blastocysts with fully enclosed inner pluripotent epiblast, you have two lineages are being set aside very early on as distinct lineages, not as in here where you start with a unilaminar blastocyst. And I think that this is actually important because we can now make a connection with imprinting, because I think that evolution of blastocysts is linked with imprinting and placental viviparity. I'll tell you about it in a second. And I think the blastocyst, evolution of the blastocyst marks a change in the strategy for vertebrate development. And I'll develop this idea a bit further later on. So I think blastocyst is a unique structure with distinct inner cell mass and trophectoderm. So these are pluripotent cells in the trophectoderm. And we know that for these cells, the maternal genome, the imprinted maternal genome is important, relatively speaking. And for these cells, outer cells, the paternal genome is relatively important. So we can now see the distinction between this kind of blastocyst and this, where you have very set out, very distinct lineages. I think one of the things which is very striking, and we don't know much about what happens in marsupials, but one thing which is really striking about Eutherian early embryos or blastomeres is the high regulative capacity. They're highly regulative. And the other thing is that you only need about 3 cells, epiblast cells, to make a whole mouse. The estimates is that you only need 3 cells to make a mouse. You may use more, but you can make a mouse with just 3 epiblast cells. So this was the experiment that actually adds to this idea as well, this distinction, parental genome, functional differences, and this, the old experiment, the start of work that Dow Salter and we did. Paternal genome, we get better trophoblast and maternal duplication embryo, but very poor trophoblast and yolk sac. In fact, Richard Gardner and I did a very elaborate micromanipulation experiment, taking the inner cell mass, parthenogenetic inner cell mass, surrounding it with primary endoderm from a normal embryo, and also surrounding it with normal trophectoderm. So you make reconstituted blastocysts, and even under those circumstances, you could not rescue parthenogenetic inner cell mass. So I think what I want to do next is to go into the germline, because this distinction that we see, epigenetic distinction we see between parental genomes, is actually occurs in the germline, and we have to really get into the fundamentals of how the germ cell lineage gets established and how it develops, to really get an understanding of the germline itself, but also understand how imprinting is linked with this. So one of the things that is interesting is that the imprinting cycle has been incorporated into the mammalian germline cycle, which is quite different from other vertebrates. It's become part of the germline cycle. So what this depicts, which is actually the same in mouse, so very similar, this happens to be human. So here imprints are introduced, the gametogenesis, create a totipotent zygote, and then you have the first epigenetic resetting event here, when the embryo starts to develop to the blastocyst stage. And this epigenetic resetting event, I think, is important for generation of pluripotent cells in the inner cell mass. So that after the embryo implants, here in humans, you start to get formation of primordial germ cells around day 16 in human. You start to get the nascent germ cells formed, and then they start to migrate, and they migrate past the hindgut, and then start migrating, and they get into the developing gonads around day 6, week 6 of the human development. You have DNA demethylation, imprints erasures. So this is a very critical epigenetic resetting event. And of course, it's actually designed to erase imprints. But you can also see that this erasure will also take out epimutations, because it doesn't discriminate between what are imprints and epimutations. This is a big advantage for eutherian mammals, that you clean up the epigenome. So if you want to think about imprinting and benefits, because we think about negatives because we have monoallelic expression and it's all bad. But what this does, it gives us something very positive, because you have to clean up the epigenome. You have to erase, and so the imprints are erased in the germline, but the somatic cells keep the imprints, so they get transmitted into the soma to adulthood. So this is the kind of the cycle. And this event here is crucial for what is germline, which is immortal. It has to go on from one generation to the next. And I think this is a big advantage. And it's critical also because you have development in utero, so you have a very high cost, maternal cost. So you clean up the epigenome to give the embryo that will develop the best chance to develop. I'm just going to go through some other things that I think that are also quite relevant and important. So I'm going to talk about this bit here, what happens when you start to form blastocysts. So as I mentioned, you have this, it's the first epigenetic resetting event for the formation of the epiblast, and you have loss of methylation here as well. So you raise DNA methylation that have come from these gametes. You have involvement of 5-hydroxymethylation and so on. There are a number of people who have worked on this, and there's a very detailed description of the mechanisms involved. The imprints are protected during this erasure because the epigenetic asymmetry is important to keep it, and you have to maintain it. So there is protection for that. And to create this blastocyst, which I said is a unique entity, and that's what gives us an advantage. So having created these two kind of lineages and created this sort of a system where you have highly regulative blastomeres and so on, I think the system is then used for cell selection and purification of epiblast cells before gastrulation. So throughout this period here, epiblast goes through very rigorous quality control, the selection. There are some papers have been published to show that the selection is occurring, and aberrant cells are eliminated, including those with low expression of T-dependent pluripotency genes, with mitochondrial mutations, aneuploid cells, and so on. So there is selection. So this, again, is kind of an advantage that you create by eliminating cells which are not very fit for future further development. So you get rid of them. And the actual number of epiblast cells is also being regulated. So if you double up the number of epiblast cells, the numbers will, there are mechanisms to ensure that you end up with the right numbers of cells. If you have half the number of cells, epiblast cells will divide faster and make up the loss, or they will eliminate some cells if there are too many. So you can see here that there are some potential advantages of this, having created this sort of system of imprinting and creating two lineages, inner cell mass and trophectoderm. This may be how the system then uses this as an opportunity to regulate the pluripotent cells, which are going to be used for development of the cell-line germline. So now I want to really come to the germline, and this is what we've been working the last 20-odd years now. So germline somatic segregation occurs in early post-implantation development. And I told you about the epigenetic resetting and so on that takes place during this period. And the germ cells get specified in this proximal epiblast. And by around day 7.5, you have approximately 30 cells. They're the founder primordial germ cells. Now, when we started looking at this, initially we were shocked to find that at the time we didn't know anything about the genetic basis for how germ cell specification occurs in mammals. There was no information about genes and what was actually happening, how the germ cell lineage was set up. What was known was that you could recognize these cells, germ cells, by TNAP is a histochemical marker. So you could recognize germ cells by a histochemical stain. There was no information about what genes or what the mechanism was, how these cells were set up. So this was the experiment that we did, was to isolate this fragment of about 300 cells, which would contain about 30 primordial germ cells, and did a differential screen using single-cell cDNAs. This is published, I won't go into this. This Michinori Saito was in my lab, and he did this work with Sheila Barton. So this was, I think this is one of the key experiments which opened up the field of mammalian germline biology. And the first gene we found was a gene we called Stella, and we thought this was the key gene, but it turned out not to be. But it has been very useful as a marker. But following on from this, we found that there were 3 genes that are crucial for germ cell specification: PIDM1, BLIMP1, PIDM14, and T52C. So we were starting to get a handle on the genetic basis for germ cell specification. There's got a lot of work involved here, but to summarize, what this tripartite genetic network does is that it represses somatic programs, it represses somatic genes, it induces expression of germ cell genes and also pluripotency-specific genes which continue to be expressed in germ cells. And for the purpose of this talk, it also initiates the epigenetic program that I told you about, epigenetic resetting. So this network is also what starts up the resetting in the germline. And so the PGC and initiation epigenetic resetting are tightly linked. And we know quite a bit about the mechanisms involved, and just to give you a glimpse of some of the mechanistic aspects of how the resetting occurs. Of the 3 genes, BLIMP1 and PIDM14 play quite a crucial role in initiating this resetting. So for example, they induce expression of some of the histone demethylases, and so there is erasure of H3 lysine dimethylation mark, and this is prerequisite for DNA demethylation to occur. You also have repression of VHRF1. So both PIDM1 and BLIMP1 are involved in repressing the 2 VHRF1 promoters. And this VHRF1 is important because it maintains DNA methylation. Without it, DNA methylation cannot be maintained. And so if you don't have VHRF1, you have replication-coupled dilution of 5-methylcytosine. In other words, the germ cells start to lose DNA methylation. So this is quite important. So this is all set up by the network, which is specifying PGCs and initiating this program. The TET enzymes are also involved, TET1 and TET2. So when you get specification of germ cells, you start to see increase in 5-hydroxymethylation, and then gradually you lose that, and you also lose 5-methylcytosine. And this you can see it at the level of a single gene, DAZL. It's quite an important gene involved in germ cell development. And you can see here transient increase in 5-hydroxymethylation, and then decline in 5-methylcytosine, and also in hydroxymethylation. So we know that there are mechanisms operating to get rid of DNA methylation. So during this time we have also X reactivation occurring, imprints erasure, global DNA demethylation down to very low levels, extremely low levels of DNA methylation. So here, starting from specified germ cells, migration, getting the gonads, very little methylation left. And there are parallel redundant mechanisms operating to ensure that you raise imprints and also DNA methylation generally. And in female embryos, you get X reactivation. So we know quite a lot about what happens in mice, so why bother about humans? And the thing is that there are developmental differences. Although the pre-implantation development in mice and humans look very similar, there are molecular differences in how this early part of development occurs. There are differences in gene expression and so on, so it's not identical. But then there's this big difference here. In rodents, you have this egg cylinder. Most of the mammals, you have bilaminar disc. So the arrangement of the early embryo between rodents and non-rodents is different. So we thought we need to look at humans' germ cell lineage, but of course, the prediction is that specification occurs around here, around day 17. Of course, we can't work on human embryos like we can on mice. So this is where we started to exploit human stem cells to try and develop methods. And this was really work, all the credit should go to Naoko Irie. She did the very first experiment, again trying to get at the genes that are important for specification of the human germ cell lineage. So this had a reporter, Nanos3, it's a very specific reporter for germ cell lineage. And there are a whole number of models that have been used. I won't go into that. But the overall idea is to try and get insight into the origin of human primordial germ cells. Where do they actually come from? How do these cells gain competence for germ cell fate? This is a question that we are interested in, still working on. And what's the mechanism of human primordial germ cell specification? And I think we've made quite a lot of progress, and I'm just going to summarize some of the key things that we have found. So when Naoko first looked at the mechanism of germ cell specification, one of the big surprises was that in humans, SOC17 is the key gene for germ cell specification, which was really completely unexpected, because SOC17 classically is known for specification of endoderm cells. So here is a gene that is involved in specification of somatic cells, not just endoderm, but other lineages as well, suddenly appears here. And it's not a minor player, it's the key player for human germ cell specification, along with PRDM1 and T52C. So we can see that compared with mice, there's no SOC17. Some of the other genes are there, PRDM14 is there, and also in human, but in humans it doesn't play a very major role. So some of the genes that are involved in mouse germ cell specification have some minor role. And so the key difference between human and many mammals is this bilaminar disc versus egg cylinder. And now people have looked at germ cell specification in a number of mammals, and it does actually turn out that these mammals, pig, rabbit, monkeys, marmoset, they all make this bilaminar disc, and they use this network. And mouse and rat, they use this one. We don't know why, but there is this divergence, evolutionary divergence. So the SOC17 is a key regulator of human primordial germ cells and any other species, including rabbits. PRDM1 is actually a direct target of SOC17, but PRDM14 in humans has a minor role, but has a very major role in mice. So there's quite a change. So we've been exploring a whole lot of things, really trying to get this in a lot of detail, trying to understand how this whole process is working. identifying some of the enhancers that are involved in the regulation of SOX17 expression. There are 3. And that we know from this kind of CRISPR interference type experiment that this enhancer 1 over here is really quite important, and then the enhancer 3 has a slightly less of a role. But they all have a role in the expression of SOX17. As we try to understand how SOX17 expression is regulated, is to really get it close to the mechanism. And as I mentioned also that SOX17 activates PRDM1, the second gene. So PRDM1 has this enhancer and the promoter. They have this SOX17 motifs. And what we did was to mutate these SOX17 motifs. So we wanted to show that PRDM1 is actually a direct target of SOX17. So if you do this experiment, luciferase experiment, if you express SOX17, you get this expression here as judged by the luciferase activity. If you have a mutation in the enhancer that we have introduced, you have reduced activity. If you have a mutation in the SOX17 mutation in the promoter, you have very little activity. But if you mutate both enhancer and promoter, you really lose activity. So this shows that SOX17 binding to PRDM1 enhancer and promoter is actually driving this expression. And what we also found is that SOX17 and BLIP1 are sufficient and necessary for human germ cell specification. If you take competent cells and co-express SOX17 and BLIP1 without any cytokines, no BMP or anything like that, you will induce germ cells. So this shows that these two are really the key players in inducing germ cells specification in human, and possibly in other mammals where you have this particular network. So I told you there's this reset at the beginning of the germ cell of the specification. So we can think of this as a kind of a reset switch. Wolfred Tang in the lab did a very comprehensive study of this. It's just now been published. So this really is the reset switch. This is a reset switch for life, because this is what keeps the germ cell lineage going. I won't go into the detail, but again, what this network does is induces expression of early germ cell genes like nanos, SOX17, KELA4, and so on. It represses somatic genes like SOX2, EMS, FOXA, HOX genes, which are not necessary, so they don't get repressed. And it initiates epigenetic reprogramming by turning on TET2, some of this demethylase, ARID5B, KDM, and so on. So again, you can see how this specification and the reset are so closely linked. So we have been trying to get at this question of what is happening after specification as the germ cells are migrating into the gonads. And again, they reach this ground state here when around week 6 or 7 in humans. There's very little DNA methylation left. But the mechanism is not the same, because what we know is that SOX17 continues to be expressed during this period. And this, amongst other genes, is actually critical for this resetting to continue. We're only scratching at the surface at the moment, but we think that this is quite an unusual involvement of a gene that is involved in somatic lineage that is doing this very fundamental job in the germ cell lineage. We know, for example, that as soon as you induce germ cells in humans, you get an increase in 5-hydroxymethylation. So these are nascent PGC-like cells made from stem cells. And this, by week 7 in vivo, you can see that 5-hydroxymethylation is very low, and you also lost 5-methylcytosine. So something is happening here, which then ends up here with very little methylation by week 7 of germ cell development, while this demethylation is going on. So we think that there are these very early changes that are taking place, but how do we get to this point? This is quite difficult, because a lot of the work we have done here so far have been trying to get hold of ex vivo primordial germ cells and analyzing them. This is good because we can understand what's going on in vivo, but they're very difficult to do mechanistic studies. We can get snapshots of what's going on, but just one more example, also been looking at histone modifications as well. So, for example, loss of 5-methylcytosine by week 8 or 9 is then replaced by histone H3 lysine 9 trimethylation. And for some reason, the olfactory receptors are protected. They don't lose the epigenetic marks. There are some loci which are called escapees, and olfactory receptors are amongst these. And that's a good reason for it, because the way the olfactory receptors are recruited in the olfactory neurons, one at a time, makes sense that they are protected, they retain this repressive mark. There are some genes that upregulate H3K4 trimethylation. This is an activation mark. You have bi signal here. And this is done stepwise, because to turn on these genes, which come on later, like DASO, PADM9. So the key point is that as you are erasing this DNA methylation, you don't get suddenly upregulation of lots of genes. The program of germ cell development is continuing. It's quite remarkable if you think about it. There are mechanisms that are turning on these genes at the right time, and this come on late as the germ cells are getting into the gonad. So it's quite an exquisite system. It's just amazing. And some genes, there are these zinc finger genes in early germ cells, human germ cells, that also get upregulated. We don't know what the function is. But these genes acquire this mark, activation mark. So some of these genes get turned on. So there's a lot to understand. It's quite a black box. We don't know what actually, kind of getting little bits of glimpses of what's going on. And then TET2 is important. I won't go into a lot of details here, but there are these genes here. If you look at them, they acquire this TET2 binding in active promoters in week 7. So these are late, and they acquire this TET2 binding. So these are genes like DASO. Some imprinted genes like PEC3, MEC3 also acquire this. And you can see expression of some of these genes coming up at this time, at week 7. So TETs are playing an important role on top of the other things that are going on. So a complex system, very difficult to get a handle on mechanisms, because there are also different players doing different things, and all very crucial and critical. So there are 2 ways to go forward from here. One is that we try and develop in vitro models, and this is not easy. So germ cells first pass through the hindgut region. So we made hindgut organoids and made primordial germ cells and put them in this hindgut organoids to see what happens. In fact, there is some impact on germ cells in the presence of hindgut. There is something coming from this hindgut organoids because this is the passage of the germ cells, they're migrating. So something is present here which we don't know yet, but there is an impact on germ cells. And then they go through into the gonads. And so there are lots of attempts to try and make synthetic, there's some success reported by some groups where you can make gonadal cells from ES cells, and you can make this kind of germinoids, kind of combine germ cells with these synthetic cells, gonadal cells, to see if you can get an impact. So the main purpose of all of this is to develop a system like this that you can manipulate and try and get at the mechanisms, because without that, we can't do it. I think we can't really do in ex vivo germ cells are fine for getting basic information, but we can't actually manipulate them. So we need to make something like this work, and this is a tough challenge because the culture times are very long. Anything that requires a very long culture period, there are lots of challenges. So this is one way of doing it. Another way of doing it is do this, use animal models. And I think rabbit can be a good surrogate model for early human development and the germline, because rabbits also use these 3 key regulators. And they do show many features of epigenetic resetting we see in humans. With a gestation period of 32 days or 30 days, this could be a way we can get a handle on mechanism in how the germ cell lineage works in rabbits, which might be applicable to humans. So this is another way of trying to understand some of the really fundamental things that are going on at this time. So I think this is the end now, and so I think I told you about the germ cell lineage. Imprints are put on, and then you get this first resetting event, which is very important for the formation of the pluripotent cells. And the epiblast cells have remarkable properties of being highly regulative, and you can actually do selection, so that's a big advantage. Germ cells are specified here around week 2, and then they start to undergo DNA demethylation. We have some idea of some of the changes that are occurring. And then there are some genes, like the olfactory receptors, that are protected. They don't, they retain some of the repressive marks. Okay, so this is summarizing what I've told you. So I think genomic imprinting actually marks significant change in the strategy for vertebrate development. This is what I believe. Because the parental genomes with epigenetic asymmetry and functional differences are essential for totipotency in mammals. And I told you about how the blastocysts, Eutherian blastocysts, with distinct inner and outer cells is a crucial innovation. That's not how the marsupial embryo looks like. And we don't have much information about what's happening in early marsupial development. This very strong quality control of epiblast cells before gastrulation, so you're taking out aberrant cells, and there's purification. And there are 2 reprogramming events, germline and blastocysts. And that parental genomes, although we have understood their role in terms of innosomas and trophoblasts, I think they probably have differential roles in many other tissues, including brain. And I think there is room for further work in this. So I think imprinting is, in a sense, is an enforcer of functional cooperation between parental genomes during development. And I don't think we understand really how they work together. I think we know bits and pieces, but we don't really know if they work together and how crucial this is and what the advantage of this kind of system is. And I think that it may have a far more important role than we have actually realized so far. So I think this is the end. I want to thank, this is my current group members, funders. They're the fantastic group of alumni I've been very fortunate to have. You'll hear from Anne here, Louis Lefebvre, many others. So it's been a fantastic journey and a lot of amazing colleagues who came and worked. Thank you very much.
Thank you, Azim. That was really good. Okay. Any questions? Sorry, the front.
Yes, very interesting. I like the model of the selection of blastocysts, and again, I think that fits with things like congenital mosaicism, where you can have trisomy in the placenta that gets rescued as disomy in the embryo, but then sometimes that's cases like Prader-Willi. But I'm wondering too about sex differences. So clearly the PGCs have two different sexes, and that is part of that. And there's some interesting, selection ideas that the sex at conception is not always the sex at birth in terms of ratios when you look at like large human populations. Have you thought about that at all in sort of the selection model? For if there, or do you think about sex differences of the embryos?
So if I understand you correctly, you're asking about difference between male and female germ cells, is that part of it?
Yeah, or the embryos that are different sexes. Do you think there's some selection, sex-based selection?
Yeah, it's a good question. So, but I think all the events that occur in the germ cell lineage until they get into the gonads occur the same, whether the embryos are male or female. The differences arise later on with sex determination, which occurs around day 13 in mice, a bit later in humans. But that's when you start to see the real differences. So yeah, there is the influence of the gonadal cells, but that comes later. So it depends on whether they're XX or XY. And then they start to become different. So you get the female germ cells entering into meiotic arrest quite early on, whereas the male entering into mitotic arrest. But this is then the influence of granulosa cells in females and the Sertoli cells in males. And that's a very interesting area of research, and there are people working on that. So you have mechanical forces involved in this as well, and also the environmental factors that induce changes in the germ cells. But that's sort of occurs pre-meiotic, and then later on you start to see further changes. But the changes that I've described, the earlier changes, they occur the same. They are not affected by whether the embryos are male or female until they get into the gonad.
So in the human cell culture, there's really no difference.
Sorry?
In the human cell culture, where it's pretty early,
Yeah.
You don't see any difference between male and female.
No, not at this point.
Yeah, great.
At least not detectable.
Thanks.
Just behind you, there's Robert. Sorry, Robert Schitzer. Can you just introduce yourself for the virtual audience?
Robert File from Montpellier. So Azim, very interesting that you compared early marsupial development with the pre-implantation development in eutherians in relation to the maintenance of imprints. So I wonder whether the idea would be that in eutherians, because of that early specification of the inner versus the outer cells, whether that would give more opportunities to maintain imprints, or whether it would give opportunities to maintain imprints differently in the cells that will give rise to the inner cell mass versus the trovectoderm.
Yeah, it's an interesting question. I think we're only talking about associations right now. We don't really, the cause effect is difficult to work out. There is something missing. I always feel that there's something missing here. I think I'd like to really know. There's been a lot talked about in terms of how imprinting affects the placenta and the trophectoderm. Because I think a lot of the debate has been on resource allocation and resource acquisition, things like that, based on the conflict idea, which is quite a good one, but not so much on the other part of the embryo, which is the inner cell mass. I don't think there's as much attention paid to that, and I think that's, to my mind, is much more interesting because that's going to give rise to the embryo and the germ cells and so on. And we don't actually know to what extent imprinted genes actually influence the way the inner cell mass develops and the competition and the selection and so on. I just don't know. But I just think that there's something missing. We're missing something, and I don't know what it is. I think we have to kind of think about it slightly differently and maybe not even think about it from the point of view of individual genes, but to look at holistic impact on each of the lineages of how imprinting contributes to that.
Irene Nechin from Paris. So, in terms of evolution, imprinting is present in much earlier in yeast, and then it disappears, and then it reappears after. What is the hypothesis for that?
Yeah, I think imprinting is seen in many, imprinting in different forms is seen in many organisms, of course, in insects as well. But I think we are thinking about what is happening in mammals. So there's a much broader question about what it does in other organisms like yeast and insects and so on. And that's kind of a talk for another time, I think. I think if you can understand what's happening between monotremes and eutherians, we might be able to understand some aspects of how the system evolved amongst mammals.
Thank you.
Thank you. I think we better call this session to an end now before the next one. Thanks ever so much.